Hi everyone. This is a quick screen capture series of slides to show you just some ideas about how to build systems with an abstraction hierarchy using devices and parts. This activity is set up with one of the bioprimers that ends with the question, what biology would you build and why? There are lots of cool things to build for medicines and fuels, and it is all very promising, but sometimes the idea of how to build these things is obscure or hard to understand. So some of what synthetic biology tries to do is add some tools to standard genetic engineering. This is a slide that you may have seen if you've looked at some of these other videos already, which adds the tools of synthetic biology like standardization and abstraction and DNA synthesis to the suite of tools that genetic engineering brought us nearly a generation ago, which includes recombinant DNA and DNA sequencing and PCR to amplify many copies. These additional tools might allow us to build complicated things like this fancy tree that grows into a house, as opposed to the tools of genetic engineering, which let us do some modifications, for instance, make plants maybe different color or less drought sensitive. So focusing on the tool of abstraction, the abstraction hierarchy that we've looked at, and this is a human-made construct, so it may be right, it may be wrong, but one way to think about the system that we're trying to build is a plain language system at the top of this hierarchy, and that system might be described as being made of different devices. And if we can describe those devices, then we can unpack what is within each device as a series of parts, and those parts are encoded by DNA. So this seems pretty obscure. One example of this abstraction hierarchy at work might be the arsenic detector, which was an iGEM project a few years ago, where a drop of well water might be added to a bacterial solution, and that bacterial solution would turn red if there was arsenic contamination in that water. So that's the system level description. Moving down a little bit lower on the hierarchy is the device level description, where you might say we need a device that senses arsenic, and then we need another device, a black box, that will produce the red color in the solution when arsenic comes in. And the benefit of having two devices instead of just one is that now if we wanted arsenic to come in and we wanted to make the solution turn green instead of red, we would swap out the second device and not have to replace the whole system. And then parts could be imagined or found in nature that could build those devices, and those parts would be made of DNA or sometimes RNA. This sounds great on paper. The question is then, how might you apply it to a system that you would want to build? And one sort of straightforward system that we can use just to begin to look at this is from another iGEM project from Melbourne. This is the introduction to their project by the Melbourne team. I'll play just a little bit of their Jamboree presentation. Oops.
Hi guys, my name is Craig and this is Elisa. We represent Melbourne University. For you guys who don't know where Melbourne is, it's on this biggish island over here at the other side of the world from where we are right now. It's in the southern hemisphere, and things are a bit different down there. For example, we had the iGEM working period, which starts in July, happens to be winter instead of summer. And right now, the Jamboree dates happen to be right on the exam period. That explains why we only have two of us right here. Our team is a bit bigger, but some of them couldn't make it because of the exams. That guy you see floating there is actually the founder of this team, but he did all the looking for funding and lab space and all that stuff, but during the first week of the lab period, he mysteriously disappeared from us. We couldn't get contact from him at all, and a month later, we got an email saying that he got hospitalized and he couldn't continue the team anymore. But anyway, what he initially intended the team to do is to expand on the red light receptor system that was created by Texas a few years ago. and make a system in which something happens when you produce another light beam, and you at this intersection point you have an output which, when you remove the lights, it stays there. But we were not sure what this output would be.
So we're developing our output, and we're thinking of having a flotation device using gas vesicles. So these are protein encapsulated gas vesicles, and you can see this transverse section of this bacterial cell is a matrix, and all these little circles of vesicles full of gas. And if you take a closer look at it, you can see that we have these protein-bound cylinders with sort of conical ends, and these are completely impermeable to anything other than gas. So they fill with gas and cause the bacteria to float. So now that we have our flotation device, we're quite fond of both ideas, so we thought we'd put them together. So we have our vat full of buoyant bacteria, a response to red light, response to blue light, some way to integrate them together in an AND gate, and we thought we'd make our final output adhesion, where so we get a clump of bacteria forming nowhere else but in the intersection of these two light paths.
All right, so that's just the introduction that they had to their iGEM project, and I'm going to interrupt them there so that we can take this system that they just described for us and try to see how we might build it through that abstraction hierarchy with devices and with parts. So the system is shown on the left, and I'm just going to start to list what devices we would need in order to build it. So the things that they listed are, among them is a red light sensor, which is shown at the top. You also need a blue light sensor. You need some mechanism for the cell to know when it is sensing both of those things. So we're going to call that an AND gate, something that will integrate the information from both of those sensors. And then there were a couple of output devices, things that they wanted the cells to do once they had sensed both the red and the blue light. They wanted the cells to float, so they needed a flotation device. And then they also wanted the cells to adhere to one another. So an adhesion device, adhere device. And then one thing that they hadn't mentioned in the video that I showed you so far is that they also had an output which was green fluorescent protein, which would happen when the cells were floating and adhering to one another, so that you would know that everything was working because the cells would start to glow green. So these 6 devices are the ones that they would like to build in the system. This is a list of devices that we would need. And so now what we can do is think about connecting them and building them into a cell. So I'm going to draw this big rectangle as our cell. And one of the first things that we know that we're going to have to put into this is the red light sensor, which I'll call RLS. We also need the blue light sensor. And we need the information from both of those sensors to go into an AND gate, something that tells the cell that both things are happening. And then the output from that AND gate, we're going to need it to be adhesion. We're going to need it to be flotation, FLT. And then, as I said, the other piece that they didn't mention, but that is true, we also need it to be green fluorescent protein. So this is one way to think about wiring up the cell so that it has these components within it that are going to carry out the program that you are interested in. One thing that's a little ambiguous from that iGEM description is the timing of the flotation versus the adhesion. So maybe what you're thinking is that maybe you knew that you needed the red light sensor and the blue light sensor, and that both of those are going to go into an AND gate. And maybe when the cell detects both of those, it will adhere. The cells will start to adhere to one another, and the cells will also start to produce GFP. but maybe there is a constant level of expression of the flotation device. That is, maybe in this vat, the cells are just constantly floating, and that only when they sense the blue light and the red light do they need to adhere and make GFP. And so these are two different wiring diagrams. They have the same devices within them, but they would be implemented in different ways. And so by drawing this out, you can start to think, well, what do I really want my cell to do? Do I want the information, do I want them to stay adhering to each other even when we remove the red light and the blue light? Can the red light and the blue light come in sequence, or do they have to come at the same time? These are all design questions that you can start to think about in a concrete way by drawing out your cell with a design level specification like this. And then you can imagine, well, GFP, we know where to get GFP in terms of sequence and how to make cells express GFP. So we can do that. Oops, I see that I've got a typo here. That should be GFP. All right, so that's a reasonably straightforward system for thinking about your system in terms of devices and then how you might break these devices down into parts. This bioprimer on biodesign does a little bit of an introduction to that, and I'll make two last points before ending this short video. One is that a nice outcome from working in biodesign like this is that you start thinking about these questions of engineering, how you would design, build, and test your system. But there is also a scientific process in here that by testing your ideas, you can test hypotheses about the cell and how the cell will actually work. So I'll leave you with one more thought, and that is about a more complex, maybe a more dynamic system, which was developed way back in 2004 by the very first pre-iGEM team, which was here at MIT during the independent activities period. They called their system the Polkadorks, and I have a small movie here that shows how they wanted their system to work. So the system overview, rather than telling you in words, is to show you this dynamic system where each of these black dots is a cell on some plated media. And the idea is that at some point, one of those black cells will randomly turn to red. When it does, the red cell starts to emit something, this green something, which then attracts other cells to it. When the cell, I'm going to see if we can, when the cells migrate towards that red cell, they get there. When the red cell turns back to black, it no longer is emitting the green thing, the green substance, and the other cells swim away. And the idea would be that over time, different cells will turn red, attract the collection of cells that are nearby, and so you'll have dynamic pattern formation over the Petri dish over time. So this much more dynamic, more complex system than the one we looked at from Melbourne can, in fact, be described through the device systems parts framework that we were thinking about. And I'll just show you briefly that the IAP team did in fact give a device level system diagram to describe their Polkadorks system. They further broke those devices down into parts, so you can see within this framework now there are arrows to indicate promoters and green ovals to indicate ribosome binding sites, purple arrows for open reading frames, and then these red circles for transcriptional terminators. So each of these devices can be broken down into parts that you can source through standard biological parts or through nature by looking at all the genomic sequencing that's available. And then you can lay these out into almost a breadboard style and know which of these devices and the parts within them need to signal to other parts. So that is the extent of what I wanted to show you just as a framework for building systems through devices and through parts. It's very instructive to go ahead and try to think of a system of your own and try to think about which devices and parts you might need to build it, and not to get too hung up with the idea that they may not be available right now to you. You could probably go into genomic databases and find the parts that you would need. So good luck and have fun.